Well, thank you very much to the organisers for the invitation to talk today. I'd like to review a little bit about uniQure, who we are, and then talk about some of our recent data that we announced last week, and a little bit more about some of our pipeline projects. We are a public company, so our disclaimers. So uniQure is a company with a long history of innovation. It's been around for over 25 years. We had the first approved gene therapy in the Western world. I think we have 2 approved AAV therapies now, hoping for a third. We had the first commercially licensed gene therapy manufacturing facility, and we had the first AAV-delivered gene silencing approach for Huntington's disease. And this is the project that I'd spend most of our time today talking about. With our AAV5 platform, we also demonstrated with HemGenix, a product that's licensed for hemophilia B, that it is effective even in patients with pre-existing neutralizing antibodies. And that was approved by the FDA in 2022 and EMA in 2023. And today we'll focus on Huntington's disease, where we got the first RMAT for treatment for Huntington's disease. Our pipeline is illustrated on this slide. Hemophilia B, as mentioned, we have a product called HemGenix. This is licensed to our partner CSL Behring for worldwide commercialization. And we have 4 clinical programs behind that: AMT-130 in HD, a genetic form of amyotrophic lateral sclerosis, or Lou Gehrig's disease, the superoxide dismutase 1 mutation, Fabry disease, and a program for mesial temporal lobe epilepsy, as well as a preclinical pipeline. But today, I'll talk in more depth about Huntington's disease. It's a autosomal dominant disease, devastating, striking patients in midlife, typically in their 40s and 50s, at the most productive stage of their life. Early symptoms include cognitive and motor and functional psychiatric behavioral disabilities, to the point that after about 15 years, the patients become unable to support themselves and unfortunately pass. It is characterized by particular vulnerability to the median spinal neurons in the striatal nuclei, the caudate putamen, and you can see the enlargement of the ventricles at the expense of the neurons in the striatum. The approach that we're taking is, with AMT-130, is it's a AAV5 delivered microRNA. This is an engineered microRNA that's designed to endogenously in the cell produce effectively an antisense targeting exon 1 of the HTT gene, so it targets not only the full-length Huntington protein, mutant Huntington protein, but also the increasingly recognized HTT1A splice variant, which is thought to be particularly toxic in patients and expands its presence with somatic instability, which is where the CAG repeat has been demonstrated to get larger in particularly vulnerable neurons. We administer this by intraparenchymal administration, so it's 3 injections per hemisphere into the caudate and putamen nuclei. So it's a precision delivery to those regions of the brain that require it. So the idea here is to minimize systemic exposure, so we use effectively a very low dose relative to some of the more broader systemically delivered AAVs. And as I mentioned, it is addressing all forms of the toxicity caused by all known forms caused by the HTT, and it can be broadly performed in centers. The clinical measures are, we'll be talking about the composite UHDRS score. This is the United Huntington's Disease Rating Score. It's a composite of 4 different clinical scales that represent the core clinical features of Huntington's disease. And I'll also show you in detail a little bit about the functional capacity endpoint, which is a key secondary. We also measure neurofilament light as a measure of neurodegeneration. Neurofilament light is a protein that's released upon neurodegeneration. It's more like the accelerator on a car. As you speed up, you get the degeneration, you get more NFL released into the CSF and plasma. I won't be talking particularly about the safety profile today, but we saw no new safety profiles, and we talk a little bit about the regulatory alignment with the FDA. So the data. Last year we presented 2-year data, and we've followed this up this year with our 3-month data, showing that on the composite UHDRS score, we showed approximately a 75% decrease in disease progression compared to an external matched control. I'll talk a little bit about this external control later, but this is a large, we've used multiple external controls. This is the Enroll HD from CHDI, and it's an approach that we've been used called propensity score matching, which is aligned with the FDA as a measure to be able to compare to external controls. We cannot take patients out for 3 years and beyond in a placebo-controlled study, where for such neurodegenerative diseases where we want to be treating early, to show that in a placebo-controlled study. We also showed, excitingly, in the key secondary endpoint, total functional capacity, also a statistical significant decrease in progression relative to the external control. This slide shows the directional consistency of the data set, showing that in the cognition as well as the motor measures, so the SDMMT and the Stroop word test are cognition tests, and the TMS is a motor scale. These are the other core domains that are affected in Huntington's disease. We also showed large suppression in the expected disease progression over the 3-year period. In addition, we showed, using NFL as a supportive biomarker, that at the 3-year time point, we maintained statistical significance below baseline. So the measurements of neurofilament light as a measure of neurodegeneration was lower 3 years later than when the patients entered the study. What I haven't shown today is the low-dose group. The low-dose group did show some promising effects, not as consistent, indicating our interpretation that we think something's happening there, but it's not as pronounced as the high dose, so there's a clear dose dependence. In terms of our next steps, we're very focused as a company on bringing this to patients as quickly as possible. At the end of last year, we aligned with the FDA that our ongoing phase 1/2 study could form the basis for a BLA submission under an accelerated approval pathway, and that we wouldn't need to do another study to support that. We could use an external control as the control group, and that the CUHDRS would be the primary clinical endpoint, with the neurofilament light as a supportive evidence for therapeutic benefit, very much following the pathway that the Alzheimer's field has driven. In addition, beginning of this year in Q1, we spoke to the FDA about our manufacturing plan. We are leveraging our platform with Hemgenix, so we use a very similar AAV5-based manufacturing platform, and this allowed us to move forward with a proposal for a single PPQ batch with the drug testing, release, and quality proposals, including the potency assay. In terms of the statistical analysis plan, we aligned with the FDA in Q2. the 3-year data with the CUHDRS and propensity score matching, using the propensity score matching as the primary analysis, but also supportive propensity score weighting and other statistical methods which show remarkable directional consistency, would form the basis of the accelerated approval request, and that the Enroll HD, similarly using the Track HD and the Predict HD datasets as additional sensitivity analyses, would be used from a control basis. So in terms of our next step, we are a relatively small company. We're very much focused on this right now. We've announced that we will be holding a pre-BLA meeting to align on the complete BLA package in Q4 with a view to file with a request for priority review at the beginning of next year. In the last few minutes, I would just like to very quickly go over a couple of our other clinical stage programs. AMT-260 is also in the clinic for temporal lobe epilepsy. It's the most common form of epilepsy. There's hundreds of thousands of patients. The approach that we use here is analogous to our Huntington's program. We use engineered microRNAs to knock down a subtype of a glutamate receptor, the gluK2 gene encoding the kainate receptor, again using a direct focal injection, this time into the hippocampus. We initiated dosing in Q4 2024 with a 3+3 type design with expansion into additional patients and into other temporal lobe epilepsy pathologies. We released earlier this year the data from the first patient. Despite being on multiple anti-seizure medications, this particular patient was averaging approximately 5 to 7 seizures per month. And what we can see for the first 5 months post-dosing was a significant, over 90% suppression of the seizure activity. And we are continuing to follow this patient as well as other patients and look forward to further data release next year. One of our other clinical stage programs is in Fabry disease. This is also leveraging the AAV5 platform. We dosed our first patient last year, and this is a pure enzyme replacement approach, IV delivered, very much analogous to the approach that we took with Hemgenix for hemophilia B. While enrollment is ongoing, I can show you for the first cohort that all the patients have come off enzyme replacement therapy, and we achieved supraphysiological expression of the alpha-gal, the enzyme that's mutated in this patient group. And in fact, you can see right at the bottom, the purple line above the x-axis, that is the physiological levels. So you could say we overshot the alpha-gal levels compared to where we thought we'd be, and these levels have been maintained despite the enzyme replacement withdrawal. The lyso-Gb3 levels so far remain constant, and we continue to dose additional cohorts and follow these patients for longer time points. So in terms of our corporate objectives, we, as I've indicated, we've focused very much on Huntington's disease as our next inflection point. We have initiated the BLA readiness activities with alignment with the FDA on the key clinical, statistical, and CMC requirements, and we're looking to hold the pre-BLA meeting in Q4. In terms of the other projects, we would plan to release additional data for both the temporal lobe epilepsy and Fabry programs next year, as well as initiate new IND submissions for additional projects. The Huntington's disease was quite an inflection, quite exciting for patients and for the company. It's allowed us to raise an upsized $300 million public offering with nearly $350 million a couple of weeks ago. This will be used to fund our commercial build out of our US commercial operations to support the filing and the launch of AMT-130 in the US. And we also increased our non-dilutive funding with refinancing some debt.
Thank you,
So
uniQure.
I'll leave that there for my last slide and say thank you very much.
Thank you, uniQure.